Thanks everyone for being here. It's a great pleasure to start our second day of this meeting. And the first presentation today is going to be by Roman Zubarev from the Karolinska Institute, who is going to talk about chemical proteomics. Please.
Thank you very much. Good morning, everyone. So chemical proteomics, what is it about? Well, it's about helping people to develop drugs, and we know that there are about 20,000 prescription drugs approved for marketing. It's quite a lot. Most of these drugs, what they do, they bind to proteins, targets, and we know that very well. The problem is that the drugs are not very specific. It is very hard to create a specific drug. Most often they're not drug target many different targets and at different concentrations. And most of these targets are not intended to be targeted, and they create side effects or may create side effects. And therefore, it's important to know where, which proteins the drug targets. And then, of course, it's important to know how it is targeting. And here comes to help chemical proteomics. People bring us new molecules they find by phenotypic screening, by other methods, and we do our magical tricks. And after that, we tell them what protein is being targeted, or proteins, and binding site, the mechanism of action, the mechanism of cellular death, if this is anti-cancer drug, and so on. How do we do that? Well, here is a very busy slide, and I would like your attention to it because it may explain to you something you didn't know before if you are not an expert in proteomics, so in mass spectrometry. This is how proteomics works. So first of all, we start with tissue cells or bodily fluid, and the proteins are being extracted. Then you have a protein mixture. The SS bonds need to be reduced and alkylated to stabilize them, and then we perform enzymatic cleavage, and the proteins being fragmented into peptides. And now we, more often than not, we label them with so-called tandem mass tag. Tandem mass tag labels free amines, which are found in lysine residues as well as N-terminine. And then we have a mixture of peptides. By the way, these tandem mass tags, they created in such a way that they compose of two parts. One part is easily detaching in the gas phase. It's called reporter ion. And the other part is a balance. So the isotopic composition is tuned such a way that the total mass is the same, but the masses of reporter ions are different. So you can have 6-plex, you can have 10-plex, you can have 16, 18-plex, and the latest announced this month is that 30-plex. So you can mix together 30 different proteomes and analyze them at once. And so then we use chromatography to separate peptides. Here, every peak in the chromatogram is actually illusion of hundreds of peptides at the same time. They are being ionized by electrospray ionization, and mass spectrum is taken by a mass spectrometer. You see this is M over Z scale, and every peak here is a different peptide. And the instrument looks at the zooms in and looks at the so-called isotopic distribution of the molecular ions, and sees these isotopic peaks. The mass difference between these peaks is 1 Dalton, but on the mass over charge ratio scale, the difference is 1/3, and therefore this is 3+. Okay, so that's also looking at the position of the so-called monoisotopic peak. The mass spectrometer figures out that it must be a peptide, and then isolates this peptide in the gas phase and fragments by one of the available fragmentation techniques, and that gives fragments from which a sequence of the peptide can be established, as well as the reporter ions in the low mass region. So you see here different peaks So the relative abundance of the peak corresponds to the relative abundance of that peptide in a given sample. So you can have, as I said, up to 30 samples mixed together, and then you get relative abundances for 30 different samples. But after the peptide sequence is established, it's mapped on the protein sequence, and you can have sequence coverage on average between 30 and 60%. And that's how we do identification and quantification. And when everything is said and done in proteomics, the results are presented typically in form of a volcano plot. The X scale is the relative difference in the abundance of the protein, and the vertical scale is a minus logarithm of the p-value, that is statistical significance. And the most interesting proteins are the outliers. They are outliers in terms of the size of the difference of the abundance. On the one hand, on the other hand, the vertical scale gives the statistical significance. So here in the upper right corner, we have proteins which are upregulated in sample versus control. And in the left corner, we have proteins which are downregulated in sample versus control. If we have to compare more than two samples, and that happens very often, we use so-called OPLS-DA, and OPLS stands for orthogonal projection to lattice squares or partial least square. There are two schools of thoughts. Both agree that the abbreviation is OPLS, and then discriminant analysis. How you do that? Well, you compare one sample, one treatment against all other samples, and then every dot represents a protein, and the proteins on the right are the ones that are specifically upregulated in that treatment, and the proteins on the left are specifically downregulated. It's a very powerful technique. So let's see, we have a drug. We need to determine the target of it. Here is one method which we call FITEXP, or functional identification of targets by expression proteomics. Say this drug kills the cells. So this is an anti-cancer drug. And first thing we do, we establish so-called LC50, the concentration at which 50% of the cells are dead after, say, 48 hours. And so that's very important. And then we typically would do several cell lines and several drugs, but for each cell line and for each drug, we will use the concentration corresponding to LC50. And then we take these cells, and the cells can be still attached to the bottom, so they're still surviving, or they can be floating on the surface, that they could be dying, but the result will still be the same. We do proteomics experiment, expression proteomics analysis, and then we look at the proteins which are specifically upregulated or downregulated in response to a given drug. And the features of this method is the following. In late apoptosis, that's after 48 hours when half of the cells are dead, at LC50 concentration, drug targets change their abundance up or down very significantly. This change is exceptionally strong. This behavior is common for both surviving cells, which are still attached to the bottom of the plate, and dying cells, which are already floating belly up on the surface of the media. This behavior is common for most types of cells that are sensitive to the drug. So this is quite a powerful method, and to give you a few examples how it works. So we use 56 different drugs and look at the behavior of the protein called DHFR, dihydroxyfolate reductase. And what we see is that in response to every drug, the abundance changed very little and usually down-regulated, except for these two drugs where it shoots up quite remarkably, the abundance of DHFR. And guess what? The DHFR is the target for these two drugs. There is another drug or another protein, topoisomerase 1, and we see its response in terms of abundance change to 56 different drugs, and we see it goes up and down. There seems to be no rule, but when it goes down very exceptionally, it is the target of these three drugs. Of course, when we compare different treatments, we use OPLSDA method, the specificity increases. For instance, here the drug is vincristine, and you will see these proteins are downregulated. And these proteins are tubulins, and vincristine is tubulin polymerization inhibitor. And here we have BAP15 proteasome inhibitor, and these green proteins are proteasomes, and so on and so forth. So this method works very nicely, and it works also the single cell level. Here we treated the cells with methotrexate drug and did the proteomics analysis after 3 hours, 6 hours, 12, 24, and 48 hours. And we looked at the behavior of the cells. We knew that half of the cells are going to die after 48 hours. The question is, when does the cell decide whether they're going to live or die? And we found some proteins which are specific to this decision. And then we can differentiate the two groups. You see red and blue, and you see already after 6 hours, you can see the separation of the drugs. But the target of methotrexate, DHFR, it doesn't change its abundance very significant, but it does changes it, say after 12 hours, it's already halfway up. And after 48 hours, it is a big outlier. So this method works at a single cell level. But we never trust one single method. We always use 2 or 3 different methods complementary, because every method has a blind spot. So alternative method we're using is called thermal proteome profiling. And here's Misha Savitsky, the first author on that science paper, this method was described. And basically, it is based on the observation that proteins, when you thermally treat them, when you increase the temperature, the proteins tend to unfold. And when they unfold, they lose their solubility. And then you can, for every protein, if you measure the abundance at different temperatures, you can have so-called melting curve. To increase the specificity after treating at the given temperature, you need to spin down, centrifuge the sample to let the proteins precipitate. But anyway, you're measuring the abundance, and then you have this curve, and you do it 2 times, with and without the drug. And look at the proteins, they change their melting curve, and melting curve can be changed in the positive way, that is increase or decrease. Either way, it actually in this particular example that they show the temperature decreased. So then you can say that this is probably the target of the drug. This method works nicely, and it's totally orthogonal to phytexp. So you can do 2-dimensional analysis, and you see this is a protein, thymidylate synthase, which is a target of Tomodex, and it's a big outlier in both dimensions, in the dimension of phytexp and the dimension of thermal proteome profile in TPP. So you can bet your farm and your horse that this is a target of that drug. The only problem with TPP is that you need to measure 10 different temperature points. That needs a lot of analysis. So we came up with a shorter method, compressed method called proteome integral solubility alteration, PISA assay. And in PISA, what we do, instead of measuring the curve, we measure the integral under the curve. And how do we measure the integral under the curve? Well, very simply, we take these samples and put them together. And so measure all samples once in terms of the abundance change. And that works nicely, increases the throughput at least 10-fold. And you can do PISA in many more forms and fashions, because now you have high throughput. For instance, you can do it in intact cells and in cell lysate, and there is a difference. So you treat the intact cells with a drug, wait for 1 hour or 2 hours, and then thermally treat them, lyse, and then label the samples after digestion, multiplex, do the proteomics analysis. And what you see here in response to 5-fluorouracil, the stimulate synthase is a big outlier. So you know it's a target of this drug in intact cells, but and then you can also take a cell lysate and do the same experiment. And guess what? In this particular case, stimulated synthase is no outlier. So what happened here? Well, what happened here is in intact cells, the 5-FU drug is actively imported and then metabolized, and it's this metabolite of this drug that targets stimulated synthase. And it doesn't bind directly to in cell lysate when there is no metabolic processing. So that's how, by comparing these two results, the intact cells and cell lysate, you can determine whether the drug acts directly on the target or it needs metabolic processing. Using PISA, we can also measure another important parameter, namely the time how much the drug spends on the target. It's called residence time, because the drug only acts when it stays with the target. But sooner or later, the drug always leaves the target and then it's no longer acting. And therefore, of course, all things being equal, you are looking for the drug that stays with the target very, very long. And how do we do that? Well, we treat the cells or the lysate for some time, and then we wash out the drug, and then we wait another period of time, which is variable from 1.5 hours to 24 hours. And then we do PISA analysis and look at the PISA shift, the shift in the solubility as a function of time for different targets of the same drug. And what we see here in response to kinase inhibitor, panatinib, we see that this particular kinase, MAP14, yes, it binds the drug, but quite quickly, within few hours, the drug is leaving the target. And very little is left. For CDK5, which is also a kinase, there is no traces left. But then look at this. There's a lean protein, which is also a kinase. And the effect of the drug is still visible after 24 hours. So this is the prime target of the drug, the ones that is responsible for the mechanism of action, long-term mechanism of action. And of course, here, especially in ALLODD, we're interested to know where exactly the drug binds. Does it bind in the active site, or does it bind allosterically? And for that, we use hydrogen deuterium exchange mass spectrometry. And hydrogen deuterium exchange is based on the fact that some of the protons or hydrogens in the protein, about half of them, the ones that are bound to heteroatoms, they're actually labile. And if you put the protein in heavy water, D2O, these protons or hydrogen atoms will be replaced with deuterons or deuterium atoms. And then the rate of this exchange is actually different for different sites. And the very naive thinking would go that if the drug is bound to some part of the protein, that this site will be shielded, and the hydrogen deuterium exchange will not happen there. And therefore, by measuring the mass shift of different peptides, you would see whether the peptide has been protected or has been not by the drug. And that's how you can determine the drug location. That's the theory. The practice is a little bit more complicated than that. So as I said, about half of all hydrogens are labile, but these hydrogens are found either in the side chain or in the backbone. And these ones which are in the side chain, they really exchange it very, very fast. So the exchange happens fast, but the back exchange also happens fast. And mind you, we also need to digest the proteins before the analysis. So during this digestion, the back exchange often happens. But the deuterons or the protons which are bound to backbone nitrogen, they're exchanging slower, and the back exchange is also slower, and therefore they are more suitable for this analysis. Another parameter that we're using is pH. So the pH is very important for determining the rate of hydrogen deuterium exchange. At pH 7, the exchange happens very fast, but if you lower pH to about between 2 and 3, then you typically reach the lowest point. The exchange drops by orders of magnitude. And so also with temperature. And therefore, if you want to freeze hydrogen deuterium exchange and no longer experience back exchange, what you do, you lower the temperature and you lower the pH. So called quenching the hydrogen deuterium exchange. Of course, some exchange happens also at these conditions, but orders of magnitude slower than at normal conditions, normal temperature, normal pH. But then the problem is that you need to digest proteins that are at pH around 0.2 or 3. And trypsin, for instance, doesn't work. And trypsin is beloved in proteomics in 90% of time, maybe more than 90%, maybe 95, 99% of time we use trypsin. But trypsin likes pH 7, so it doesn't work as pH 2. And therefore, we have to use pepsin. Pepsin is one of the few proteases that still work at lower pH. And it's not as specific as trypsin. The peptides that are produced, they are typically shorter. And as I said, there's less specificity. Therefore, the mass spectra look more complicated. And therefore, first, you need to do mass spectrometry without any hydrogen deuterium exchange to establish the peptide identity. And therefore, you cannot really do hydrogen deuterium exchange at the level of the whole proteome. The system you analyze should be simpler. Typically, it's one protein and one drug, or maybe two proteins. That's it. So then you have to monitor how the mass of the peptide shifts with the time when you put it in heavy water. And then you still have to identify this peptide. You see the isotopic distribution becomes broader, and it may start to overlap with another peptide. So this makes the analysis more complicated. But then everything said and done, you would have this kind of plot where you see sequence coverage, or typically more than 80% of the protein sequence is covered by the peptides, and then you have this masses shifts at different time of hydrogen deuterium exchange. And then you do this kind of plot 2 times with and without the drug, and you look at the color change. The regions that change their color would be the ones where the drug binds or where allosteric modifications because of drug binding would take place. So you see here, for instance, this one, this system was determined by HDXMS, and this is the position of the drug. The area around it has shifted, but you don't have a resolution like in X-ray crystallography or NMR, where you can determine the exact position of binding down to a single residue. Here you cannot. And also you see there is allosterically changed site, which is far away from the binding site. But that's the reality. And you have to do more experiments to determine, to narrow down this site where the drug is really bound. The good news, however, is that hydrogen deuterium exchange works when there is no crystal, and it works for a relatively small amount of the sample. There is one emerging technique which also has structural resolution, which is based on limited proteolysis in its classical form, provided by Paolo Picotti, and here is a Nature Protocol paper, and they have a bunch of other papers. They use 2 proteases. One protease is proteinase K, which is very unspecific, and it just cleaves a lot of bonds, of course, not those protected by the drug. And then after the proteome is about half digested, you stop proteinase K, and then you use trypsin, and then you get tryptic peptides and semi-tryptic. And if you change the drug concentration and do this kind of analysis, then you can see how the protection changes for different peptides, and you can sort of determine the position of the drug binding. The problem with this method is that it doesn't have even this resolution that we have with hydrogen deuterium exchange. it gives you about 1/3 of the protein where the binding takes place. And again, it cannot distinguish by itself between allosteric and orthosteric bindings. It's one thing, but the beauty of it is that it works at the level of the whole proteome. So you determine the drug target and roughly the position of binding at the same time from the one single analysis. What we are doing, what Bogdana is doing in our group, is trying to improve this method, getting rid of protein SK and using a different approach, but I leave it to her to describe this method. So let me come to the conclusions. The conclusions are chemical proteomics reveals drug target without chemical modification of the drugs or need to create a chemical probes. One method is called FitExp, and we created an online tool called ProTarget Miner anyone can use to analyze the data. They use expression level changes for drug target convolution, can be used at a single cell level. TPP and PISA perform target convolution based on the solubility changes. These are orthogonal to expression proteomics. PISA allows one to determine the residence time of the drugs, very important. Hydrogen deuterium exchange determines the site of ligand binding. And finally, limited proteolysis is perspective methods of chemical proteomics that also provides some degree of structural resolution. With that, I would stop and welcome your questions.
Thank you very much for this very interesting review on the state of the art. Is there any question? We have time for one question. Okay, then I have one, and my question is about yesterday during the session dedicated to computational methods, we have seen many methods based on simulations. It is my understanding that especially HDX and molecular dynamics can go often together. What's your opinion? What's your...
Yeah, that's right. And actually, there are a few groups that work towards determining the structure of proteins based on HDX MS data and using computations. The problem, I talked at length with people working there. The problem there, they need a very good first approximation. And the hope was that AlphaFold in its latest reincarnation would be providing a good first approximation, and then that can be refined using HDX data. So yes, I'm very excited. I think this is very perspective method.
Okay, thank you. So let's thank Roman again.